The initial cellular and molecular events underlying memory in normal and Alzheimer's brains. A scale-accurate animation. What is memory? How are memories stored in the brain? And how do neurodegenerative diseases affect this process? Although our understanding of memory is far from complete, over the past few decades, we have gained a basic understanding of some of the underlying mechanisms involved at the cellular and molecular levels in hippocampal CA1 neurons. These mechanisms involve communication between nerve cells, neurons, within the brain. Information in the brain exists in the form of electrical signals called action potentials, which travel along neurons. The information encoded as action potentials can be transmitted from one neuron to another via synapses, which are small connection points that allow for neuronal communication. With respect to information transfer across a synapse, the neuron sending the signal is called the presynaptic neuron, and the neuron receiving the signal is called the postsynaptic neuron. Neurons are composed of many extensions called dendrites, which receive the signals, a cell body which processes the information, and a single axon which can propagate an action potential in response. When an action potential traveling along the axon of a presynaptic neuron reaches the axon terminal, it triggers the release of neurotransmitter into the synaptic cleft, a narrow space between the two neurons. Cell surface receptors on the dendrites of the postsynaptic neuron can detect these neurotransmitters and elicit a change in the membrane potential in response, referred to as an excitatory postsynaptic potential, EPSP. When enough EPSPs from enough dendrites reach the cell body of the postsynaptic neuron simultaneously, the cell body may initiate an action potential of its own in response. In this way, information in one neuron can be transmitted to another. Introduction to LTP. One mechanism of memory formation, called long-term potentiation, LTP, involves changes in communication between neurons. During LTP, a transient high-frequency action potential in the presynaptic neuron releases, via exocytosis, a large amount of the neurotransmitter glutamate into the synaptic cleft. The detection of a high concentration of glutamate by the postsynaptic neuron triggers biochemical reactions that persistently increase the magnitude of the EPSP. This leads to a more efficient generation of action potentials in the postsynaptic neuron. In a sense, the postsynaptic neuron remembers the high-frequency event. Glutamate release, but no postsynaptic EPSP. To understand the molecular mechanism underlying LTP, let's start by examining a situation in which the frequency of action potentials in the presynaptic neuron is low, a situation in which LTP does not occur. When the frequency of action potentials in the presynaptic neuron is low, the frequency of glutamate exocytosis, and thus the resulting concentration of glutamate in the synaptic cleft, is also low. Glutamate is detected by receptor channels in the postsynaptic membrane called AMPA type, glutamate receptor channels or AMPAR. When bound to glutamate, these AMPAR channels allow sodium ions to diffuse into the cell, changing the electrical potential across the cell membrane. Therefore, when the concentration of glutamate in the synapse is low, only a small number of AMPAR channels are activated, and the change in membrane potential in the postsynaptic neuron is below the threshold at which an EPSP would be generated. Eventually, glutamate is removed from the synapse, causing the AMPAR channels to close, and the membrane potential returns to its resting level. Glutamate release, NMDAR activation, and calcium entry. Next, let's examine a situation in which the frequency of action potentials in the presynaptic neuron is high, a situation in which LTP can occur. When the frequency of action potentials in the presynaptic neuron is high, the frequency of glutamate exocytosis and thus the resulting concentration of glutamate in the synaptic cleft is also high. Many AMPAR channels are thus activated. This quantitative difference from the previous scenario is significant because it means the membrane potential will change beyond a critical threshold, causing the postsynaptic cell membrane to undergo an EPSP. Therefore, in this situation, communication between presynaptic and postsynaptic cells has been successful. However, the generation of an EPSP is only the beginning, as the events of LTP are just getting started. The EPSP in the postsynaptic cell membrane also results in the dislodging of a magnesium ion from an ion channel called the NMDA-type glutamate receptor, or NMDAR. NMDAR channels require two events to become fully open: the binding of glutamate in the synapse and the removal of a magnesium ion that blocks its channel. Now that both have occurred, NMDAR channels allow additional sodium ions to flow into the cell, just like AMPAR channels, further contributing to the EPSP. NMDAR and AMPAR channels are therefore similar in that they both bind glutamate and both allow sodium ions to enter the cell. However, there are two important differences between these two ion channels. First, whereas AMPAR channels are opened whenever glutamate is in the synapse, NMDAR channels are opened only when the concentration of glutamate in the synapse is high enough to cause an EPSP by sodium ions entering through AMPAR channels. Second, in addition to sodium ions, NMDAR channels also allow calcium ions to flow into the cell. The entry of calcium ions into the cell is significant because intracellular calcium is the trigger for LTP. Calmodulin, CAMK2 activation, and AMPAR channel exocytosis. We can think of the presence of calcium ions in the cell as electricity flowing through the wires in our home. The electricity itself is not the effector, but it is required to turn on a light, a fridge, or a computer. In a cell, calcium ions do not power things like electricity does. Instead, they activate or inactivate molecular pathways by binding to calcium-binding proteins, the most significant of which is calmodulin. Once activated by calcium, calmodulin binds and activates various calmodulin-binding proteins, of which calcium calmodulin-dependent protein kinase 2, or CAMK2, is important for LTP. Once one CAMK2 subunit is activated by CAM, the active subunit can then activate its neighboring subunits, and so on, until the entire protein is active. CAMK2, in turn, phosphorylates downstream proteins, including AMPAR channels. While AMPAR channels are present in the postsynaptic membrane, they are also found on intracellular membranous compartments. AMPAR channels continuously cycle between the postsynaptic membrane and intracellular compartments through endocytosis and exocytosis. The phosphorylation of AMPAR channels by CAMK2 inhibits the endocytosis and promotes the exocytosis of AMPAR channels, leading to increased amounts of AMPAR channels in the postsynaptic cell membrane. As the presynaptic action potentials eventually come to an end, glutamate is removed from the synapse, AMPAR channels close, the postsynaptic cell membrane repolarizes, and magnesium moves back to block the NMDAR channels once again. However, this time the postsynaptic cell has changed. There are now more AMPAR channels in the postsynaptic membrane and it is ready for action. Although LTP lasts for some time, eventually the phosphatase calcineurin dephosphorylates AMPAR, and the cells return to baseline, erasing the cellular memory. The significance of more AMPAR in the cell membrane. The increased amount of AMPAR channels in the cell membrane now means that more sodium ions can enter the cell for any given concentration of glutamate. If we repeat the first scenario, with a low frequency of action potentials in the presynaptic neuron, the postsynaptic cell can now undergo a larger EPSP because there are more AMPAR channels in the postsynaptic membrane. In other words, the same low concentration of glutamate that was insufficient to result in an EPSP in the first scenario may now be sufficient. This entire process is called LTP, and it's what allows the postsynaptic cell to remember an event and respond differently to the same event the next time around. The effect of amyloid beta on LTP. Not only are we beginning to resolve the basics of LTP in hippocampal DA1 neurons, but we are also beginning to understand how neurodegenerative diseases like Alzheimer's disease can disrupt learning and memory. One of the hallmarks of Alzheimer's disease is the formation of amyloid beta peptides, which are too small to be seen in this animation. Levels of the neurotoxic amyloid beta peptide are directly correlated with memory loss. In keeping with this, amyloid beta inhibits LTP in various ways. One example is given here. As shown earlier, in normal LTP, CAMK2 phosphorylates AMPAR, stopping its endocytosis and allowing the receptor to accumulate in the membrane. After a short time, the events of LTP stop. Part of that stoppage is the removal of phosphate groups from AMPAR. This reestablishes endocytosis and thus results in less AMPAR in the synaptic membrane. LTP then stops. In Alzheimer's disease, amyloid beta binds to calcineurin, causing it to become active all the time. As a result, the dephosphorylation and endocytosis of AMPAR keeps its level low in synapses, causing LTP to be impaired. This is believed to contribute to memory dysfunction in Alzheimer's individuals.